Let's jump right in. Welcome to This Explainer. Today, we're shrinking ourselves down to the microscopic level to explore the fascinating, incredibly rapid, and honestly, sometimes downright scary world of bacterial growth. If you've ever wondered how a single rogue cell can turn into a full-blown infection practically overnight, or how scientists harness these microscopic factories to pump out life-saving medicines, well, you're in exactly the right place.
Here's our quick roadmap for today. We'll start with the basics of multiplication. Then, we'll track the bacterial growth curve. Third, we'll look at continuous culture, basically how we sustain life. And finally, we'll dive into how we go about measuring the unseen. Let's get started.
Okay, Section 1. The basics of multiplication. Or, how exactly one becomes many.
First, we got to define what we actually mean by growth in the bacterial world, because it's a bit different than how you and I grow. For us, growing generally just means getting bigger, right? But in microbiology, bacterial growth is an orderly increase in all cell components, and it happens on two distinct levels. Yes, there's an increase in the actual size of the cell, but the real key is the increase in the number of cells. You see, before a single cell even thinks about dividing, it goes through an intense period of internal manufacturing, synthesizing new nucleic acids, building proteins, the whole shebang. Once that DNA replication is totally complete, it triggers the formation of a septum, which eventually splits the cell right in two.
Now, what's really wild about this is the sheer variation in how long this takes. We call this the generation time, which is literally just the time it takes for a bacterial population to completely double. And the scale here is absolutely astonishing. You have a bacteria like Clostridium with a generation time of just 10 minutes. That means a population can double in the time it takes you to brew and drink a cup of coffee. Then you've got E. coli at a brisk 20 minutes. But on the totally opposite end of the spectrum, you have Mycobacterium tuberculosis. Notice its incredibly sluggish generation time of 20 hours. It can take up to 2 whole weeks just for one of those cells to grow into a visible colony.
So, what actually happens when you introduce these rapid multipliers into a fresh, nutrient-rich environment? That brings us to Section 2, the bacterial growth curve and the rise and fall of a microscopic civilization.
When bacteria are dropped into a fresh batch of closed medium, their population follows a highly predictable 4-part journey. If we map this out over time, it rises and falls like a massive biological roller coaster. This standard growth curve is totally fundamental to understanding microbiology, so let's break down each of these 4 unique phases.
First up is the lag phase. When cells are first introduced to a fresh medium, they don't just start dividing instantly. Instead, they enter this lag phase, a period of quiet preparation. The population number remains temporarily unchanged, but hey, don't mistake a lack of division for a lack of activity. Even though there's no apparent cell division happening, these cells are highly active metabolically. They're furiously synthesizing enzymes, RNA, and proteins, basically packing their bags and getting totally geared up for their new home.
Once they're fully prepped, the population absolutely explodes into the exponential or log phase. In this phase, the cells are dividing regularly by binary fission, and it's a geometric progression. 1 becomes 2, 2 becomes 4, 4 becomes 8, you get the idea. They're dividing at a constant maximum rate, meaning their generation time hits its absolute minimum. This right here is the peak of bacterial productivity. And here is why this rapid growth window is absolutely crucial for medicine. Because the bacteria are metabolizing so fast and rapidly synthesizing cell walls and proteins, they are incredibly vulnerable. So if scientists want to perform antibiotic sensitivity testing to figure out which antibiotics will actually work against an infection, this log phase is exactly when they prefer to do it. But in a closed environment, this massive boom just can't last forever. Eventually they hit the stationary phase. Think of this as a true period of crisis for our microscopic civilization. Growth is severely limited because all the available nutrients in the environment are completely exhausted. And to make matters worse, toxic waste products and sometimes even naturally produced antibiotics begin to build up all around them.
Now here's a really fascinating detail. The stationary phase is not completely static or frozen in time. The overall viable count of the bacteria stays totally level, but that's strictly because a grim equilibrium exists. The exact rate of dying cells perfectly matches the rate of newly formed cells. They're still desperately trying to divide, but they're dying off just as fast.
Finally, the harsh environment can no longer support even that fragile equilibrium, which triggers the death or decline phase. During this phase, the number of viable cells rapidly decreases geometrically. It's essentially the exact reverse of the explosive log phase from earlier. The culture is completely collapsing.
But life finds a way, right? If you take those struggling survivors from the decline phase and move them into a fresh, nutrient-rich medium, a totally routine lab process known as subculturing, they enter a readjustment phase. The bacteria acclimate to the fresh environment, and this entire epic 4-part cycle starts all over again.
So what if scientists don't want the culture to die? What if they want to keep a culture permanently trapped in that highly productive log phase? Well, that brings us to Section 3, sustaining life through continuous culture, or basically, cheating the growth curve.
To pull this off, microbiologists use a brilliant continuous technique. Instead of a closed batch where nutrients eventually run out, they keep the culture volume and cell concentration perfectly constant. They do this by constantly piping fresh, sterile medium into the vessel at the exact same rate that they drain away the spent medium, which carries off the waste and excess cells. By perfectly balancing this input and output, the rate of newly produced cells exactly equals the rate of lost cells.
This magical balancing act is usually handled by a sophisticated device called a chemostat. It's the most widely used continuous cultivation system in the world. By maintaining a constant culture volume, it effectively prevents the bacterial population from ever hitting that stationary crisis phase. This is a real game changer because it allows for the industrial-scale production of vital things like insulin, enzymes, and other bioproducts.
So, we understand how they grow, and we know how to sustain them indefinitely. But practically speaking, how on earth do microbiologists actually count organisms they can't even see? Let's move to Section 4, measuring the unseen.
Scientists broadly split their clever counting methods into two main buckets: direct measurement and indirect measurement. With direct methods, scientists are literally counting the physical cells using specialized microscopes or electronic sensors. But with indirect methods, they aren't counting the original cells themselves, but rather something the cells produce, like the visible colonies they form when they grow on a plate.
Let's check out the direct methods first. One popular tool is the Petrov-Hauser chamber. It's essentially a special glass slide with an incredibly precise microscopic grid ruled onto it. You put a drop of bacterial suspension under a microscope and manually count the cells inside the tiny squares. It's fast, and you can actually observe the shape of the bacteria. Then there's the electronic particle counter. Here, bacteria pass through a microscopic hole. As a cell passes through, it increases electrical resistance, generating a tiny signal that a computer counts. It's unbelievably fast, but there is one massive flaw. The machine just counts physical particles. It cannot tell the difference between a live, healthy cell and a totally dead one.
To solve that dead cell problem and get a true viable count of only the living bacteria, scientists rely on indirect plate counting. Imagine a petri dish filled with solid agar. A liquid sample of bacteria is mixed in, and once the agar solidifies, the unseen individual bacteria are trapped right in place. Over time, each single living cell multiplies geometrically until it produces a visible white dot, which is a colony. By just counting those visible colonies, scientists know exactly how many living bacteria were in the original sample.
And you know, beyond just counting individual numbers, microbiologists can also measure growth by assessing total cell mass or cell activity. For cell mass, a super common method is turbidimetry. Scientists use an instrument called a spectrophotometer to measure how cloudy or turbid a liquid culture is. Simply put, the cloudier it is, the more cell mass you have. You can also measure the nitrogen content, or literally just dry the cells out and weigh them. On the flip side, you can measure cell activity by tracking specific chemical changes, like gauging exactly how much acid the bacteria are pumping out as a byproduct of their metabolism.
Which brings us to a final thought for today. When you realize that a single microscopic cell can become billions in a matter of mere hours, you really have to ask, how might our ability to endlessly harness this rapid growth completely change the future of biomanufacturing and medicine? We are essentially commanding an invisible, tireless workforce.
This explainer has merely scratched the surface of microbiology and the wild mechanics of bacterial growth. So, keep wondering, keep questioning, and keep exploring the fascinating, invisible empire all around us. Thanks for joining me on this microscopic journey.